Okay, protein synthesis is just a fancy term describing how DNA is turned into proteins. Understanding DNA is highly essential to understanding this video. So if you want to up your DNA game, I can highly recommend this truly amazing video that will brush away those mental cobwebs. Otherwise, protein synthesis. Let's get into it. Here we have a cell in the human body. This is the nucleus, this is the cytoplasm, and this is the cell membrane. Cells also contain something else. They contain proteins. Different types of proteins are found all over the cell and are crucial for the cell to work correctly. Every day, cells have to make thousands of new proteins. Now, proteins are long chains of something called amino acids folded up into a 3D structure. Luckily, the cells in our body are amazing and have a well-established process to make new proteins. Our cells have something called DNA, deoxyribonucleic acid, which is a blueprint for a protein. So we have this, and we want to get to this. How does our cell manage to do that? The answer is protein synthesis. The first thing to know is there are two stages to protein synthesis. The first stage is called transcription, and the second stage is called translation. Naturally, let's look at transcription first. Let's zoom right in on an animal cell so that this is the cell membrane and this is the cytoplasm. That means this is the nucleus, this is the nuclear membrane, and these little holes are called nuclear pores. Inside the nucleus itself, we have the DNA. Transcription begins as the DNA unwinds and the hydrogen bonds that hold the two strands of DNA together break. Now for a new key term, a nucleotide. A nucleotide is a phosphate attached to a sugar attached to a base. Inside the nucleotides, the bases can be one of four options: adenine, cytosine, guanine, and uracil. Wait, in part 1, we saw that DNA is made up of adenine, cytosine, guanine, and thymine. So what's uracil? Uracil is a base that actually replaces thymine in the protein synthesis process. So now we're ready to see how protein synthesis works. Ready? Let's go! The nucleotides move in between the DNA strands and pair up with their corresponding complementary base pairs. Adenine nucleotides pair with thymine, cytosine nucleotides pair with guanine, and guanine nucleotides pair with cytosine. Uracil nucleotides pair with adenine because it's replaced thymine. After our bases have paired up, the sugar phosphate backbone is reformed. The new line of nucleotides that has just been produced breaks away from the DNA strand to produce an amazing new molecule called mRNA, messenger ribonucleic acid. The mRNA is small enough to fit through the nuclear pores. The DNA is too large to fit out, which is why our cells have this ingenious system for carrying information from DNA in the nucleus out into the cytoplasm. This completes the first part of protein synthesis. This completes transcription. But that's only half the story, because now we have the second step of protein synthesis: translation. Translation occurs in the cytoplasm at something called a ribosome. Part 2 sees the introduction of a new character, tRNA, transport ribonucleic acid. This is what tRNA looks like. It's an amino acid attached to 3 bases. Different tRNA molecules will have different combinations of the 3 bases. Let's check out the role our tRNA plays. Our mRNA is in the cytoplasm, ready to be copied through the process of translation. Our new character, tRNA, descends down to our mRNA strands and lines up with the first 3 bases. If the 3 bases on the tRNA match the 3 bases on the mRNA, the amino acid is deposited. Next, another tRNA molecule moves down and the process is repeated. A match will cause the amino acid to be deposited, and it will bond to the first amino acid. This process is repeated along the entire length of the mRNA until we have a whole chain of amino acids. A chain of amino acids. That's what a protein is. This chain of amino acids is a protein. The DNA has been copied, which has led to the production of a specific protein, which is exactly what our cell needed. Which now brings us to a new, very important word: a gene. A gene is a section of DNA that codes for a specific protein. A section of DNA is just this, the codes for means converted to, and a protein is our chain of amino acids. Okay, let's work backwards to see where a gene fits into all of this. The protein is the chain of amino acids. The amino acids are coded for by tRNA, which is coded for by the bases in the mRNA. The mRNA is coded for by the bases in the DNA, so if we trace it back, we can actually see that this is a gene. It's a section of DNA that codes for a particular protein. That is it. That's how DNA is made into a protein. That's how proteins are synthesized. Anyways, thank you so much for watching. Let me know if you have any questions, and I'll see you in the next one. DNA.